Welcome back to part 2 of cells in our general science series. And we just left off, and we're going to start talking about cilia and flagella. Cilia and flagella provide motility and allow cells to move around. So cilia are like short little hairs that are on the outsides of human cells, and they kind of wiggle things around to make things move. And the flagella is kind of like a long tail, and the cell that should be most common with, or known most about that have these, are sperm cells. So sperm cells have flagella. Those are the little tails that let them move around. The structure of these cilia and flagella are basically just like a bunch of little tiny tubes that are all grouped together into one section. And here in this picture, you can see the cross section of what they look like. So they got all these tubes inside of it. Centrioles, these are microtubular structures that play a role in cell division. In the next couple episodes, when we talk about cell division, we'll talk about centrioles more. Then we're going to talk about some membrane organelles. Membrane organelles are the ones that are found in the membranes, and they do the different tasks that cells need to be done. The most important of which is the nucleus. Nucleuses are the control centers of cells. They have a double membrane because they contain DNA, which is the most important thing in a cell, is the blueprints that tell the cell what to do. So all of those things are very important. That's why there's a double layer. So like I said, that nuclear envelope separates the nucleus from the rest of the cell. It's double membrane, but there are some holes that allow things to go in, mostly proteins that are going to help the cell interpret DNA. DNA is what's found inside the nucleus, and it's our hereditary material. It has all the instructions on how to make us and then how to function. That DNA is built into chromosomes, and it's what allows us to build proteins, and it also allows us to divide our cells. The Nucleolus is what helps direct the synthesis of RNA. The RNA, of course, is the instructions to build proteins. Endoplasmic reticulum helps move substances within cells, so they're kind of like the highways that allow things to move around. And there's two different types: a rough and a smooth one. Some cells have rough, some cells have smooth, some have both. The rough endoplasmic reticulum, those are where ribosomes are attached, and this is where proteins are manufactured. Not all ribosomes are attached to this rough ER, but a lot of them are, and they sometimes modify proteins from the ribosomes. The smooth endoplasmic reticulum has no attached ribosomes, and these mostly have enzymes that help build molecules. So this is where like carbs and fats and other types of proteins are made. The Golgi apparatus, that's involved in the synthesis of the plant cell wall, and it also is kind of like the packaging and shipping station of the cell. So that's where things get like placed and packaged and then sent off to different areas. Like I said, the Golgi apparatus functions kind of like this. Molecules will come in vesicles, which are like little trucks that are being transported through the cell. The vesicles are going to fuse into the membrane. The molecules might be modified a little bit, and then they're pinched off into a separate vesicle, and that vesicle leaves and goes off to go do whatever it's going to do. Here's kind of a picture of something called exocytosis. Exocytosis is how cells get rid of trash. So for instance, here's the nucleus, the rough ER, and the smooth ER. You're going to build a protein. That protein's going to go to the Golgi. And then if that protein needs to leave the cell to go elsewhere, it's going to go over here and be exocytosis out of the cell. Lysosomes contain digestive enzymes, and they function in several different ways. They can aid in cell renewal, so if something gets damaged in the cell, lysosomes will help fix that. They break down old cell parts so they can be reused and used in new proteins or new pieces. And sometimes they'll be used to digest invaders. So if something gets into the cell that's not supposed to be there, a lysosome will destroy it by eating it. Vacuoles are storage sacs. They're more common in plants than animals, but animals do have them sometimes, and they contain water or food or waste or basically whatever. They're just storage areas for all the different things the cell might need. Some of our bacteria, like organelles, they release and store energy, and there's two types. There's the mitochondria, which release energy, and chloroplasts, which store energy. Mitochondria have their own DNA because this is probably one of the other more important organelles in a cell. Mitochondria is the one that allows cells to create and then use energy. So mitochondria, they break down fuel molecules, basically glucose and sometimes fatty acids, and they release energy, which is in the form of ATP, which is a giant molecule that can be broken apart to create energy. Chloroplasts, on the other hand, only exist in plant cells, and they are derived from photosynthetic bacteria, so they've come from bacteria, and they are solar energy capturing organelles. So this is why plants are green, because they have chloroplasts, and they capture sunlight and they turn that sunlight into energy. They do that through the process of photosynthesis, and it basically makes sugar out of sunlight, which is pretty nifty. Wish humans could do that, it'd be really useful. Here's some review. We're just going to skip through that part. And now we're going to talk a little bit about molecule movement and cells. So there's a couple different ways for molecules to move in cells. There's passive transport, active transport, endocytosis, and exocytosis. And we're going to talk about each one in detail. Passive transport is when there's no energy required. And that's basically where things move into a cell or around a cell due to a gradient. So that means that there's a difference in concentration or pressure or charge, and so things will move to equalize the gradient, right? If there's a lot of something on one side, it might move to the other side to even things out. There's several different types of passive transport, diffusion, osmosis, and facilitated diffusion. Diffusion is basically the idea that molecules are going to move to equalize concentration. And you would see that if you were maybe like making lemonade and you put sugar into the water and you put it in as a cube, that cube's going to dissolve and spread out over time. Osmosis is a special form of diffusion, and that's that fluid flows from a lower solute concentration to another one. So if you pour two different liquids together and they're going to mix, they're going to slowly mix until they're equally distributed. We're going to skip over this one right here, and this one. Facilitated diffusion is about a differentially permeable membrane. So sometimes these channels help molecules or ions enter or leave the cells, but there's no energy used. It's still diffusion. It's just being helped along by these special proteins that are allowing things in and out. This is how that would work. So you've got something outside of the cell here that needs to get in. It's going to go into the transport protein and then just pop back out on the inside of the cell. So there's no energy involved in that. It's just like a tunnel. Now, active transport is where energy is required. So in this instance here, we've got some potassium up here on the outside of the cell. And what will happen is sodium will come on the other side. They're going to have to drop off some energy, which is ATP. Remember we said that ATP is the energy source of cells. ATP will lose a phosphate and become ADP. And the energy released from releasing that phosphate will allow the sodium to come out and the potassium to come in. So this is because of a sodium-potassium pump. Endocytosis is movement of large types of material, and it's always into cells. So this is like where you're moving in like a large amount of particles or organisms or really large molecules like sugar. So this is how endocytosis would work. The plasma membrane is going to just kind of surround the material. The edges of the membrane would meet, and then that would become a vesicle, which could transport all that stuff around the cell. There's two different types. There's the eating one, and then there's drinking. So this one's for liquids, and this one's for solids. Exocytosis is the other form, of course. This is when we're getting rid of something. So that's when a bunch of stuff is going to be moving in a vesicle to the wall. It's going to kind of merge into the wall and then secrete all that stuff. And there's a picture of it right here happening in a microscope. So you can see how that looks. That's exactly what I just explained. And there's a cell right there. Thank you so much for watching and learning a little bit about cells with me. Next time we'll learn about the cell cycle and how that works. Thanks again for watching. Tune in later for some more general science.